Golgi apparatus, protein modification and sorting. The Golgi apparatus is a series of flattened membrane-bound sacs called cisternae, responsible for modifying, sorting, and packaging proteins and lipids synthesized in the endoplasmic reticulum. Proteins destined for secretion or localization to specific cellular compartments undergo post-translational modifications such as glycosylation, phosphorylation, and proteolytic cleavage within the Golgi apparatus. Additionally, the Golgi apparatus plays a critical role in sorting these modified molecules into vesicles for transport to their final destinations, highlighting its importance in cellular trafficking and secretion. Mitochondria, powerhouses of the cell. Mitochondria are double membrane-bound organelles, known as the powerhouses of the cell due to their role in energy production through aerobic respiration. These organelles contain their own circular DNA and ribosomes, suggesting a semi-autonomous origin derived from endosymbiotic bacteria. Within the mitochondria, enzymes involved in the citric acid cycle, Krebs cycle, and oxidative phosphorylation catalyze the conversion of nutrients into adenosine triphosphate, ATP, the primary energy currency of the cell. The presence of mitochondria underscores the metabolic diversity and energy requirements of eukaryotic cells. Conclusion: understanding the exclusive structures present in eukaryotic cells is fundamental for grasping the complexities of cellular organization and function. From the nucleus, which houses the genetic material, to the mitochondria, responsible for energy production, each organelle plays a distinct yet interconnected role in cellular physiology. Mastery of these concepts is essential for success on the MCAT and forms the basis for further exploration into the intricacies of eukaryotic cell biology. Question 2. Cell structure. Not in the nucleus. Organelles outside the nucleus. Not all organelles are located in the nucleus. Mitochondria, for example, generate energy for the cell. In eukaryotic cells, the nucleus serves as the command center housing genetic material, but numerous essential cellular processes occur outside of the nucleus. These cellular structures, collectively referred to as organelles, are vital for maintaining cell function, metabolism, and overall homeostasis. Understanding the structures and functions of these organelles is crucial for grasping cell biology and is often tested in exams like the MCAT, Medical College Admission Test. Here are some key structures found outside the nucleus. Mitochondria. Structure. Mitochondria are double-membraned
Question 3. Organelle that recycles cellular materials. Organelle responsible for recycling. The lysosome is the organelle primarily responsible for recycling cellular materials through processes like autophagy. Organelle that recycles cellular materials, the lysosome. Introduction. Lysosomes are membrane-bound organelles found in eukaryotic cells, known for their role in cellular waste management and recycling. These organelles contain various hydrolytic enzymes capable of breaking down macromolecules such as proteins, nucleic acids, lipids, and carbohydrates into their constituent monomers, which can then be reused by the cell for energy production or biosynthesis. Structure. Lysosomes are spherical vesicles enclosed by a single lipid bilayer membrane. The acidic interior of lysosomes, pH around 4.5 to 5, is maintained by proton pumps located in the lysosomal membrane, which actively transport protons into the lysosome. This acidic environment is crucial for the optimal activity of lysosomal enzymes. Function. Intracellular digestion. Lysosomes play a pivotal role in intracellular digestion by breaking down various biomolecules. They fuse with endocytic vesicles formed during endocytosis, thereby digesting the engulfed materials, such as extracellular proteins and pathogens, into smaller molecules. Autophagy. Lysosomes are involved in autophagy, a cellular process by which damaged organelles and proteins are degraded and recycled. During autophagy, cytoplasmic components are engulfed by double membrane structures called autophagosomes, which then fuse with lysosomes for degradation. Programmed cell death, apoptosis. Lysosomes also participate in programmed cell death, or apoptosis. They release hydrolytic enzymes into the cytoplasm, causing degradation of cellular components and ultimately leading to cell death. Waste recycling. Lysosomes are responsible for recycling cellular materials by breaking down obsolete organelles, malfunctioning proteins, and other cellular debris into their basic building blocks. These building blocks, such as amino acids, nucleotides, fatty acids, and sugars, can then be transported back into the cytoplasm and used for energy production or as precursors for biosynthesis. Clinical relevance. Lysosomal storage disorders, LSDs. Mutations affecting lysosomal enzymes or lysosomal membrane proteins can lead to lysosomal storage disorders. In these conditions, undegraded substrates accumulate within lysosomes, causing cellular dysfunction and tissue damage. Examples include Tay-Sachs disease, Gaucher's disease, and Niemann-Pick disease. Cancer. Dysregulation of lysosomal function has been implicated in cancer progression and metastasis. Lysosomes contribute to cancer cell survival by promoting nutrient recycling, facilitating autophagy-mediated resistance to chemotherapy, and aiding in invasive behavior. In summary, lysosomes serve as the recycling centers of eukaryotic cells, participating in intracellular digestion, autophagy, programmed cell death, and waste recycling. Dysfunction of lysosomal processes can have profound implications for cellular homeostasis and human health, underscoring the importance of understanding lysosomal biology in both normal physiology and disease pathology. Question 4. Combination of alleles is also called understanding genetic combinations, allelic interactions in genetics. Genetic combinations and alleles, the combination of alleles or different forms of a gene, determines traits like eye color or blood type. Introduction. In genetics, the combination of alleles plays a crucial role in determining an organism's phenotype. Alleles are different versions of a gene, and the various combinations of alleles contribute to the diversity observed within populations. Understanding the principles behind the combination of alleles is fundamental in comprehending inheritance patterns and predicting phenotypic outcomes. Definition of allele. An allele refers to one of the alternative forms of a gene that occupies a specific locus on a chromosome. Alleles may differ in their nucleotide sequence and can result in variations in observable traits, known as phenotypic traits. Combination of alleles. The combination of alleles refers to the specific arrangement of alleles at corresponding loci on homologous chromosomes. These combinations can be homozygous, where two identical alleles are present, or heterozygous, where two different alleles are present. Terminology. Genotype, the genetic makeup of an organism, representing the specific combination of alleles present for a particular trait. Phenotype, the observable characteristics or traits of an organism, resulting from the interaction between its genotype and the environment. Dominant allele, an allele that is expressed in the phenotype when present in either homozygous or heterozygous condition. Recessive allele, an allele that is expressed in the phenotype only when present in a homozygous condition. Codominant alleles, alleles that are both expressed in the phenotype when present together in a heterozygous condition, without one being dominant over the other. Principles of allelic interaction. Law of segregation states that during gamete formation, the alleles of a gene segregate from each other so that each gamete carries only one allele for each gene. Law of independent assortment states that alleles of different genes assort independently of each other during gamete formation, leading to various combinations of alleles in the offspring. Incomplete dominance occurs when the heterozygous phenotype is an intermediate blend of the two homozygous phenotypes. Complete dominance occurs when the phenotype of the heterozygote is indistinguishable from the phenotype of the homozygous dominant genotype. Multiple alleles. Genes that have more than two alleles within a population, increasing the potential combinations of alleles and resulting phenotypes. Conclusion: the combination of alleles is a fundamental concept in genetics, influencing the inheritance of traits in organisms. Mastery of this topic allows for the prediction of phenotypic outcomes and provides insights into the mechanisms of genetic variation and evolution. Understanding allelic interactions is essential for any aspiring biologist or medical professional to comprehend complex genetic inheritance patterns and their implications for human health and disease.
Question 5. Reproductive cells in meiosis. Meiosis and chromosome division. Meiosis is the process of cell division that leads to the formation of reproductive cells, sperm and egg. Cells undergoing meiosis have half the number of chromosomes as somatic cells. Understanding reproductive cells in meiosis. Key concepts for the MCAT. Introduction. Reproductive cells, also known as gametes, are integral components of sexual reproduction in multicellular organisms. Meiosis is the specialized cell division process responsible for the formation of gametes, ensuring genetic diversity and perpetuation of species. Mastery of these concepts is crucial for success on the medical college admission test, as it tests understanding of fundamental biological principles. Key concepts: meiosis. Overview. Meiosis is a type of cell division that occurs in specialized cells called germ cells to produce gametes. It involves two successive divisions, meiosis I and meiosis II, resulting in the formation of 4 daughter cells, each with half the number of chromosomes as the parent cell. Gamete formation. During meiosis, a diploid cell, 2N, typically a germ cell, undergoes DNA replication followed by 2 sequential divisions to produce haploid gametes, N. This reduction in chromosome number is essential for maintaining the chromosome number across generations. Genetic variation. Meiosis promotes genetic diversity through several mechanisms. Crossing over. Exchange of genetic material between homologous chromosomes during prophase I leads to the reshuffling of genetic information. Independent assortment. Random orientation of homologous chromosomes during metaphase I results in the assortment of maternal and paternal chromosomes into daughter cells independently. Random fertilization. Fusion of gametes during fertilization is random, further increasing genetic variability. Phases of meiosis. Meiosis consists of 2 main phases, each subdivided into stages. Meiosis I, reduction division. Prophase I, homologous chromosomes pair up and undergo crossing over. Metaphase I, homologous pairs align at the metaphase plate. Anaphase I, homologous chromosomes separate and move to opposite poles. Telophase I, nuclear envelopes form around chromosomes. Meiosis II, similar to mitosis but with half the number of chromosomes. Prophase II, chromosomes condense and spindle fibers form. Metaphase II, chromosomes align at the metaphase plate. Anaphase II, sister chromatids separate and move to opposite poles. Telophase II, nuclear envelopes form around chromosomes and cytokinesis occurs, resulting in the formation of 4 haploid daughter cells. Gamete specifics. In males, meiosis gives rise to 4 sperm cells, each with equal amounts of cytoplasm. In females, meiosis produces 1 ovum, egg, and 3 polar bodies, with the majority of cytoplasm retained by the egg to support embryonic development. Conclusion. Understanding the intricacies of reproductive cells in meiosis is essential for success on the MCAT, as it forms the basis of many biological processes, including inheritance, genetic diversity, and reproductive biology. Mastery of these concepts empowers aspiring medical professionals to comprehend and analyze complex biological systems with precision and accuracy. Question 6. Cell division in somatic cells. Introduction to cell division. Define cell division and its importance in somatic cells. Brief overview of the cell cycle and its phases. Stages of cell division. Interphase, the resting phase before cell division begins. Prophase, chromosomes condense and become visible. Metaphase, chromosomes align at the cell's equator. Anaphase, chromatids separate and move to opposite poles. Telophase, chromosomes decondense and two new nuclei form. Significance of cell division. Why cell division is crucial for growth, repair, and maintenance. The role of somatic cell division in multicellular organisms. Implications and applications. How errors in cell division can lead to diseases like cancer. Practical applications of understanding cell division in medicine and biotechnology. Conclusion and recap. Summarize key points covered in the video. Encourage viewers to explore further and ask questions. Cell division in somatic cells. Cell division in somatic cells is a fundamental process essential for growth, development, and tissue repair in multicellular organisms. Somatic cells are non-reproductive cells that make up the majority of the body's tissues and organs. The process of cell division in somatic cells is known as mitosis. Mitosis is a highly regulated process consisting of several distinct stages: prophase, prometaphase, metaphase, anaphase, and telophase. During mitosis, a parent cell divides into two identical daughter cells, each containing the same genetic information as the parent cell. Prophase, chromosomes condense and become visible under a microscope. The nuclear envelope disintegrates, and spindle fibers begin to form from centrosomes located at opposite poles of the cell. Prometaphase, spindle fibers attach to the centromeres of chromosomes, which have now fully condensed. The chromosomes move towards the center of the cell. Metaphase, chromosomes align along the metaphase plate, an imaginary plane equidistant between the two spindle poles. This alignment ensures that each daughter cell will receive an identical set of chromosomes. Anaphase, sister chromatids separate and move towards opposite poles of the cell, pulled by the shortening spindle fibers. Each chromatid is now considered a separate chromosome. Telophase, chromosomes decondense and nuclear envelopes reassemble around each set of chromosomes at opposite poles. The spindle fibers disintegrate, and cytokinesis, the division of the cytoplasm, begins. Cytokinesis. In animal cells, cytokinesis involves the formation of a cleavage furrow, which pinches the cell membrane inward until the parent cell is divided into two daughter cells. In plant cells, a cell plate forms at the center of the cell, gradually developing into a new cell wall that separates the daughter cells. Regulation of cell cycle. The cell cycle is tightly regulated by checkpoints that ensure proper progression and prevent aberrant cell division. Key regulatory proteins, such as cyclins and cyclin-dependent kinases, CDKs, control the transitions between different phases of the cell cycle. Checkpoints, including the G1S, G2M, and spindle assembly checkpoints, monitor DNA integrity, cell size, and spindle attachment to chromosomes, respectively, before allowing progression to the next phase. Significance. Understanding the process of cell division in somatic cells is crucial for various biological processes, including embryonic development, tissue regeneration, and maintenance of tissue homeostasis. Dysregulation of cell division can lead to diseases such as cancer, where cells proliferate uncontrollably due to mutations in genes involved in cell cycle regulation. MCAT relevance. The MCAT exam frequently assesses knowledge of cell biology, including cell division processes such as mitosis and cytokinesis. Test takers should be familiar with the stages of mitosis, their characteristics, and the regulatory mechanisms that ensure proper cell cycle progression. Additionally, understanding the significance of cell division in somatic cells and its relevance to human health and disease is essential for success on the MCAT.
Question 7. How cells divide by meiosis. How many chromosomes, meiosis, and chromosome division Meiosis is the process of cell division that leads to the formation of reproductive cells, sperm and egg. Cells undergoing meiosis have half the number of chromosomes as somatic cells. Understanding meiosis, the process of cell division and chromosome count. Introduction. Meiosis is a fundamental process in biology, essential for the production of gametes, sperm and egg cells, and the maintenance of genetic diversity in sexually reproducing organisms. This highly regulated process involves 2 consecutive divisions, resulting in the formation of 4 daughter cells, each containing half the number of chromosomes as the parent cell. Understanding the intricacies of meiosis is crucial for grasping concepts related to genetics, inheritance, and evolution. Overview of meiosis. Meiosis consists of 2 distinct stages, meiosis I and meiosis II. Each stage involves a series of complex events, including DNA replication, chromosome segregation, and cell division, ultimately leading to the formation of haploid daughter cells. Meiosis I, prophase I. This is the longest phase of meiosis, characterized by the condensation of chromatin into visible chromosomes. Homologous chromosomes pair up and undergo genetic recombination, known as crossing over, which promotes genetic diversity. Metaphase I. Homologous chromosome pairs align along the metaphase plate, facilitated by spindle fibers attached to the centromeres. This arrangement ensures independent assortment, contributing further to genetic variability. Anaphase I. Homologous chromosomes separate and migrate to opposite poles of the cell, driven by the shortening of spindle fibers. Telophase I. Chromosomes arrive at the poles, and the nuclear envelope reforms around each set of chromosomes. Cytokinesis, or cell division, may occur, resulting in 2 haploid daughter cells. Meiosis II, prophase II. Similar to mitotic prophase, the nuclear envelope breaks down and spindle fibers reassemble. However, there is no further DNA replication. Metaphase II. Chromosomes align individually along the metaphase plate. Anaphase II. Sister chromatids separate and move toward opposite poles of the cell. Telophase II. Chromatids arrive at the poles, and nuclear envelopes reform around the separated chromosomes. Cytokinesis completes the process, yielding 4 haploid daughter cells, each genetically distinct from the others and the parent cell. Chromosome count in meiosis. In most sexually reproducing organisms, including humans, the chromosome count is halved during meiosis. This reduction ensures that when gametes fuse during fertilization, the resulting zygote will have the appropriate diploid chromosome number characteristic of the species. For example, in humans, where the diploid chromosome count is 46, 23 pairs, meiosis reduces this number to 23 chromosomes in each gamete. Conclusion. Meiosis is a highly orchestrated process essential for sexual reproduction and genetic diversity. Through the reduction in chromosome number and genetic recombination, meiosis ensures the production of genetically unique gametes, contributing to the variability observed within populations. Understanding the intricacies of meiosis, including chromosome behavior and segregation, is crucial for comprehending inheritance patterns and evolutionary processes in biology. Question 8. Tetrad formation. In meiosis, tetrad formation in meiosis, a crucial step in genetic recombination. Tetrad formation. In meiosis, homologous chromosomes pair up to form tetrads, allowing for genetic recombination. Tetrad formation is a pivotal event that occurs during meiosis, the process by which diploid cells give rise to haploid gametes. This process is crucial for genetic diversity, as it allows for the shuffling and recombination of genetic material. Understanding tetrad formation is fundamental for comprehending the mechanisms underlying genetic variation and inheritance. Key concepts. Meiosis. Overview. Meiosis consists of 2 sequential divisions, namely meiosis I and meiosis II, each comprising prophase, metaphase, anaphase, and telophase stages. The primary objective of meiosis is to reduce the chromosome number by half, ensuring that gametes receive a single set of chromosomes. Prophase I. Tetrad formation primarily occurs during prophase I of meiosis I. This phase is further subdivided into 5 stages: leptotene, zygotene, pachytene, diplotene, and diakinesis. Tetrad formation specifically occurs during the zygotene stage. Synapsis and tetrad formation. Synapsis is the process by which homologous chromosomes pair up and become physically connected along their lengths. This pairing results in the formation of a tetrad, also known as a bivalent, composed of 4 chromatids. Each tetrad consists of 2 sister chromatids from each homologous chromosome. Crossing over. One of the most critical events associated with tetrad formation is crossing over, or genetic recombination. Crossing over involves the exchange of genetic material between non-sister chromatids within a tetrad. This process promotes genetic diversity by generating novel combinations of alleles on the chromosomes. Chiasma formation. Crossing over leads to the formation of chiasmata, which are visible points of contact between non-sister chromatids. Chiasmata represent sites where genetic material has been exchanged, and they help hold homologous chromosomes together until they segregate during meiosis I. Importance of tetrad formation. Tetrad formation and crossing over are essential for genetic diversity and the production of genetically unique gametes. They introduce variability by mixing and matching alleles from homologous chromosomes, leading to the creation of offspring with different genetic compositions. Disjunction and reductional division. Following tetrad formation, homologous chromosomes segregate during anaphase I of meiosis I, leading to the reduction of chromosome number in daughter cells. This process ensures that each gamete receives a complete set of chromosomes with a unique combination of alleles. Tetrad formation during prophase I of meiosis plays a critical role in genetic recombination and the generation of genetic diversity. Through the process of crossing over, tetrad formation facilitates the exchange of genetic material between homologous chromosomes, resulting in the production of genetically unique gametes. Understanding the mechanisms underlying tetrad formation is essential for comprehending the principles of inheritance and evolution. Question 9. Alteration in normal gene sequence. Gene sequence alterations. Mutations or alterations in gene sequences can have profound effects, from genetic disorders to changes in physical traits. Alteration in normal gene sequence. Alteration in normal gene sequence, also known as genetic mutation, is a fundamental concept in genetics with profound implications for human health and disease. Mutations can occur in various ways, including substitution, insertion, deletion, duplication, or inversion of nucleotides within a gene sequence. These alterations can lead to changes in the encoded protein's structure and function, potentially resulting in diseases or disorders. Types of mutations. Point mutations. Point mutations involve changes in a single nucleotide base within the DNA sequence. These changes can be categorized into missense mutation, a single nucleotide substitution that leads to the incorporation of a different amino acid in the protein sequence. Depending on the location and nature of the substitution, missense mutations can either be benign or result in malfunctioning proteins associated with diseases. Nonsense mutation, a mutation that converts a codon specifying an amino acid into a premature stop codon, leading to the premature termination of protein synthesis. This often results in truncated and non-functional proteins. Silent mutation. These mutations occur when a nucleotide substitution does not alter the amino acid sequence due to the redundancy of the genetic code. As a result, there is no phenotypic change. Frameshift mutations. Frameshift mutations involve the insertion or deletion of nucleotides in a DNA sequence, leading to a shift in the reading frame during translation. This alteration can drastically change the amino acid sequence downstream of the mutation site, often resulting in non-functional proteins. Repeat expansion mutations. These mutations involve the abnormal expansion of repetitive DNA sequences within a gene. This expansion can lead to various neurodegenerative disorders, such as Huntington's disease and Fragile X syndrome. Implications of genetic mutations. Disease susceptibility. Genetic mutations can predispose individuals to various genetic disorders, including cancer, cystic fibrosis, sickle cell anemia, and Huntington's disease. Understanding the genetic basis of these diseases is crucial for diagnosis, prognosis, and potential therapeutic interventions. Evolutionary significance. Mutations serve as a driving force for evolutionary change by introducing genetic diversity within populations. Beneficial mutations may confer selective advantages, leading to their propagation over successive generations, whereas deleterious mutations may be eliminated through natural selection.
Pharmacogenomics, genetic variations among individuals can influence drug responses and metabolism. Pharmacogenomic studies aim to identify genetic factors that influence drug efficacy, toxicity, and individualized treatment strategies. In conclusion, alterations in normal gene sequences play a pivotal role in both health and disease. Understanding the mechanisms and implications of genetic mutations is essential for various fields, including medicine, evolutionary biology, and pharmacology. This knowledge is fundamental for addressing complex genetic disorders, designing targeted therapies, and advancing personalized medicine approaches. Question 10. Probability for type A blood. Probability of blood type. Understanding genetics helps calculate the probability of inheriting specific blood types, such as type A. Blood types are determined by the presence or absence of specific antigens on the surface of red blood cells. The most common system used to categorize blood types is the ABO blood group system, which consists of 4 main blood types, A, B, AB, and O. These blood types are determined by the presence or absence of antigens known as A and B antigens. In the ABO blood group system, individuals can inherit one of 4 possible blood type alleles from their parents, A, B, or O. The A allele codes for the A antigen, the B allele codes for the B antigen, and the O allele codes for no antigen. It is recessive to A and B. Each individual inherits 1 allele from each parent, resulting in 3 possible genotype combinations: AA, AO, or OO for blood type A, BB, BO, or O for blood type B, AB for blood type AB, and OO for blood type O. Let's consider the parents in this scenario. One parent has blood type A, genotype AA or AO, and the other parent has blood type B, genotype BB or BO. Since blood type A and blood type B are codominant, a child of these parents could inherit one of the following genotypes: AA, AO, BB, or BO. To determine the probability of the child having blood type A, we need to consider the possible combinations of alleles from the parents. Since both parents carry at least 1 A allele, there are 3 possible combinations that result in a child with blood type A, AA, AO, or AB. Given that one parent has blood type A, genotype AA or AO, and the other parent has blood type B, genotype BB or BO, the possible combinations are as follows: AA, blood type A, XBB, blood type B. All offspring would be blood type AB, AO x BO. AA, blood type AX, blood type B, 50% chance of blood type A, and 50% chance of blood type AB, AOXBO. AO, blood type A, XBB, blood type B, 50% chance of blood type AXBO, and 50% chance of blood type AB, XBO. AO, blood type A, XBO, blood type B, 25% chance of blood type AOXBO, 25% chance of blood type B, AOXBO, 25% chance of blood type XBO, and 25% chance of blood type O, AOXBO. So the probability of the child having blood type A, either AA or AO, would be the sum of the probabilities from scenarios 2 and 3. Probability equals 0.5 to 0.5 plus 0.5 to 0.5 equals 0.25, or 50%. Therefore, there is a 50% chance that the child would have blood type A, either AA or AO, based on the given parental blood types. This probability remains the same regardless of whether the child's blood type is indicated as 1A or A on the MCAT test, as both refer to blood type A. Question 1. Cell structure. Which structure in eukaryotic cells is responsible for the synthesis of proteins? A. Mitochondria, B. Golgi apparatus, C. Nucleus, D. Ribosomes. Question 2. Cell structure not in the nucleus. Which organelle is responsible for synthesizing proteins, modifying them, and packaging them into vesicles for transport? A. Golgi apparatus, B. Endoplasmic reticulum, C. Mitochondria, D. Lysosomes. Question 3. Organelle that recycles cellular materials. Which organelle is primarily responsible for recycling cellular materials in eukaryotic cells? A. Golgi apparatus, B. Endoplasmic reticulum, C. Lysosome, D. Peroxisome. Question 4. Combination of alleles is also called. Which of the following terms refers to the combination of alleles present in an individual? A. Genotype, B. Phenotype, C. Homozygote, D. Heterozygote. Question 5. Reproductive cells in meiosis. In meiosis, what are the reproductive cells produced? A. Somatic cells, B. Gametes, C. Stem cells, D. Epithelial cells. Question 6. Cell division in somatic cells. Which phase of the cell cycle is characterized by the replication of DNA? A. G1 phase, B. S phase, C. G2 phase, D. M phase. Question 7. How cells divide by meiosis. How many chromosomes, during which stage of meiosis do cells divide into daughter cells with half the number of chromosomes as the parent cell? A. Prophase 1. Metaphase 2, C. Anaphase 1. Telophase 2. Question 8. Tetrad formation in meiosis. During which phase of meiosis does tetrad formation occur? A. Prophase 1 and C. Anaphase or telophase 1.
Question 9. Alteration in normal gene sequence. Which of the following is an example of a frameshift mutation? A. Substitution mutation. B. Insertion mutation. C. Silent mutation. D. Missense mutation. Question 10. Probability for type A blood. Two individuals, one with blood type 1AA and the other with blood type 1BB, have a child. What is the probability that the child will have blood type A? A. 0%, B. 25%, C. 50%, D. 75%. Question 1. Cell structure. Which structure in eukaryotic cells is responsible for the synthesis of proteins? A. Mitochondria. B. Golgi apparatus. C. Nucleus. D. Ribosomes. Correct answer, D. Ribosomes. Explanation. Ribosomes are the cellular organelles responsible for protein synthesis in eukaryotic cells. They can be found either free in the cytoplasm or bound to the endoplasmic reticulum, ER. Ribosomes function by reading the mRNA, messenger RNA, and translating the genetic code into specific amino acid sequences, ultimately leading to protein synthesis. A. Mitochondria. Mitochondria are double-membraned organelles responsible for energy production through aerobic respiration. While they play a crucial role in cellular energy metabolism, they are not directly involved in protein synthesis. B. Golgi apparatus. The Golgi apparatus is involved in modifying, sorting, and packaging proteins for secretion or delivery to other cellular locations. While it plays a vital role in the processing of proteins, it does not directly participate in protein synthesis. C. Nucleus. The nucleus is the control center of the cell, housing the cell's DNA and directing cellular activities. It contains the genetic information required for protein synthesis, but it does not directly synthesize proteins. Instead, it houses the DNA that encodes the instructions for protein synthesis, which are then transcribed into mRNA and transported to the cytoplasm, where ribosomes carry out the actual synthesis. Therefore, option D, ribosomes, are the correct answer because they are directly responsible for protein synthesis in eukaryotic cells, while options A, B, and C are incorrect as they are involved in other cellular functions, but not in protein synthesis. Question 2. Cell structure not in the nucleus. Which organelle is responsible for synthesizing proteins, modifying them, and packaging them into vesicles for transport? A. Golgi apparatus. B. Endoplasmic reticulum. C. Mitochondria. D. Lysosomes. Correct answer, B. Endoplasmic reticulum. Explanation. A. Golgi apparatus. The Golgi apparatus is responsible for modifying, sorting, and packaging proteins into vesicles for transport. However, it does not directly synthesize proteins. B. Endoplasmic reticulum. The endoplasmic reticulum, ER, is responsible for synthesizing proteins, modifying them, especially in the rough endoplasmic reticulum, where ribosomes are attached, and packaging them into vesicles for transport to various cellular destinations. This process occurs via the rough endoplasmic reticulum, R, where ribosomes are attached, and the smooth endoplasmic reticulum, SER, which is involved in lipid synthesis and detoxification. C. Mitochondria. Mitochondria are primarily responsible for generating ATP through cellular respiration. While they do play a role in synthesizing some proteins, particularly those involved in their own function, they are not primarily responsible for protein synthesis, modification, and packaging. D. Lysosomes. Lysosomes are membrane-bound organelles containing digestive enzymes. They break down various biomolecules and cellular waste materials. They are not involved in protein synthesis, modification, or packaging. In summary, option B, endoplasmic reticulum, is the correct answer as it directly relates to the synthesis, modification, and packaging of proteins within the cell. Question 3. Organelle that recycles cellular materials. Which organelle is primarily responsible for recycling cellular materials in eukaryotic cells? A. Golgi apparatus. B. Endoplasmic reticulum. C. Lysosome, peroxisome. Correct answer, C. Lysosome. Explanation. A. Golgi apparatus. The Golgi apparatus is involved in modifying, sorting, and packaging proteins for secretion. It plays a role in the transportation of molecules but is not primarily responsible for recycling cellular materials. B. Endoplasmic reticulum. The endoplasmic reticulum, ER, is involved in protein synthesis, lipid metabolism, and calcium storage. While it plays a role in cellular functions, it is not primarily responsible for recycling cellular materials. C. Lysosome. Lysosomes are membrane-bound organelles containing digestive enzymes. They function to break down various macromolecules, including proteins, lipids, nucleic acids, and carbohydrates. Lysosomes play a crucial role in cellular waste disposal and recycling of cellular materials through processes such as autophagy and phagocytosis, making them the correct answer. D. Peroxisome. Peroxisomes are involved in lipid metabolism and detoxification reactions, but they are not primarily responsible for recycling cellular materials. They primarily participate in reactions involving hydrogen peroxide and fatty acids. In summary, the correct answer is C. Lysosome, because it is primarily responsible for recycling cellular materials through various digestive processes. The other organelles listed have different functions within the cell and are not primarily dedicated to recycling cellular materials. Question 4. Combination of alleles is also called. Which of the following terms refers to the combination of alleles present in an individual? A. Genotype. B. Phenotype. Homozygote. D. Heterozygote. Correct answer, A. Genotype. Explanation. A. Genotype. The genotype refers to the combination of alleles present in an individual for a particular gene or set of genes. It represents the genetic makeup of an organism, including both the dominant and recessive alleles. This is the correct answer because the genotype directly addresses the combination of alleles, which is the focus of the question. B. Phenotype. Phenotype refers to the observable traits of an organism, which result from the interaction between its genotype and the environment. While the phenotype may reflect the expression of alleles, it does not directly represent the combination of alleles themselves. This is incorrect because while phenotype reflects traits influenced by alleles, it doesn't specifically refer to the combination of alleles. C. Homozygote. Homozygote refers to an individual having two identical alleles for a particular gene, e.g. AA or AA. While this term is related to allele combinations, it specifically describes individuals with identical alleles, not the combination of alleles in general. This is incorrect because it refers specifically to individuals with identical alleles, not the combination of alleles in general. D. Heterozygote. Heterozygote refers to an individual having two different alleles for a particular gene, e.g. AA. Like homozygote, this term is related to allele combinations, but it specifically describes individuals with different alleles, not the combination of alleles in general. This is incorrect because it refers specifically to individuals with different alleles, not the combination of alleles in general.
Question 5. Reproductive cells in meiosis. In meiosis, what are the reproductive cells produced? A. Somatic cells. B. Gametes. C. Stem cells. D. Epithelial cells. Correct answer, B. Gametes. Explanation. A. Somatic cells. Somatic cells are non-reproductive cells that make up the majority of an organism's body tissues and organs. These cells undergo mitosis for growth, repair, and maintenance of the organism's body. However, they are not involved in meiosis, which specifically produces reproductive cells. B. Gametes. Gametes are reproductive cells produced during meiosis. In humans, these are sperm cells in males and egg cells in females. Meiosis is the process by which these specialized cells are formed, containing half the genetic material, haploid, of the parent cell. Gametes fuse during fertilization to form a diploid zygote, which develops into a new organism. C. Stem cells. Stem cells are undifferentiated cells that have the potential to develop into various cell types. While stem cells play crucial roles in development and tissue repair, they are not exclusively produced during meiosis. Meiosis specifically leads to the formation of gametes, which are specialized reproductive cells. D. Epithelial cells. Epithelial cells are one of the main types of cells in the body, forming the linings of various organs and tissues. Like somatic cells, they undergo mitosis for growth and repair, but are not directly involved in meiosis or the production of reproductive cells. In summary, the correct answer is B. Gametes, because meiosis specifically generates these specialized reproductive cells, with half the genetic material of the parent cell, which are crucial for sexual reproduction. Options A, C, and D represent other types of cells that are not directly involved in the process of meiosis or the production of gametes. Question 6. Cell division in somatic cells. Which phase of the cell cycle is characterized by the replication of DNA? A. G1 phase, B. S phase, C. G2 phase, D. M phase. Correct answer B. S phase. Explanation: A. G1 phase. During the G1 phase, the cell grows in size and performs its normal functions. However, DNA replication has not yet occurred. Therefore, this phase is not characterized by DNA replication. B. S phase. The S phase, or synthesis phase, is where DNA replication occurs. This phase is crucial for the duplication of genetic material in preparation for cell division. C. G2 phase. In the G2 phase, the cell continues to grow and prepares for mitosis, or meiosis in reproductive cells. While important cellular activities such as protein synthesis occur during this phase, DNA replication has already occurred during the S phase. D. M phase. The M phase, or mitotic phase, is where cell division occurs, including both mitosis and cytokinesis. DNA replication has already occurred during the S phase, so this phase does not involve DNA replication. Therefore, the correct answer is B. S phase, as it is specifically characterized by DNA replication, a critical process in cell division. Question 7. How cells divide by meiosis? How many chromosomes? During which stage of meiosis do cells divide into daughter cells with half the number of chromosomes as the parent cell? A. Prophase 1, B. Metaphase 2, C. Anaphase 1, D. Telophase 2. Correct answer C. Anaphase 1. Explanation: in meiosis, which is the process of cell division that produces gametes, sperm and egg cells, the number of chromosomes in the resulting daughter cells is halved compared to the parent cell. This reduction is crucial for sexual reproduction to maintain the correct chromosome number in the offspring. Anaphase 1 is the stage in meiosis 1 where homologous chromosomes, each consisting of two sister chromatids, are separated and pulled to opposite poles of the cell. This separation ensures that each daughter cell receives one member of each homologous pair, leading to the reduction of chromosome number by half. Therefore, during anaphase 1, cells divide into daughter cells with half the number of chromosomes as the parent cell. Let's examine why the other options are incorrect. A. Prophase 1. Prophase 1 is the stage where chromosomes condense, the nuclear envelope breaks down, and homologous chromosomes pair up and undergo genetic recombination, crossing over. While important for genetic diversity, the actual division of cells into daughter cells with half the chromosome number occurs later in meiosis. B. Metaphase 2. Metaphase 2 is the stage in meiosis 2 where chromosomes align along the metaphase plate, but the division of cells into daughter cells with half the chromosome number has not occurred yet. Metaphase 2 is where sister chromatids are separated, ensuring that each daughter cell receives one chromatid from each chromosome, but the chromosome number remains the same. D. Telophase 2. Telophase 2 is the final stage of meiosis 2, where nuclear envelopes reform around the separated chromosomes, and cytokinesis occurs, resulting in the formation of 4 haploid daughter cells. However, the reduction in chromosome number occurs during anaphase 1, not telophase 2. Telophase 2 is responsible for the physical separation of the chromosomes into distinct daughter cells. Therefore, the correct answer is C, anaphase 1, as it is the stage where cells divide into daughter cells with half the number of chromosomes as the parent cell. Question 8. Tetrad formation. In meiosis, during which phase of meiosis does tetrad formation occur? A. Prophase 1, B, C, anaphase 1, D, telophase 1. Correct answer, A. Prophase 1. Explanation: tetrad formation, also known as synapsis, occurs during prophase 1 of meiosis. This is the stage where homologous chromosomes pair up and exchange genetic material through a process called crossing over. Tetrad formation involves the alignment of homologous chromosomes side by side, creating a structure known as a tetrad or bivalent. This alignment facilitates the exchange of genetic material between the homologous chromosomes, promoting genetic diversity. Thus, prophase 1 is the correct answer. Option B. Metaphase 1 is incorrect because during this phase, tetrads align at the metaphase plate but do not form. Homologous chromosomes align independently on the metaphase plate, each attached to spindle fibers via the het. Option C. Option C. Anaphase 1 is incorrect because this phase is characterized by the separation of homologous chromosomes, not the formation of tetrads. Homologous chromosomes are pulled to opposite poles of the cell during anaphase 1. Option D. Telophase 1 is incorrect because it is the final stage of meiosis 1, where the separated homologous chromosomes arrive at opposite poles of the cell and the nuclear membrane may reform around them. Tetrad formation occurs prior to telophase 1, during prophase 1. Question 9. Alteration in normal gene sequence. Which of the following is an example of a frameshift mutation? A. Substitution mutation. B. Insertion mutation. C. Silent mutation. D. Missense mutation. Correct answer, B. Insertion mutation. Explanation. A frameshift mutation occurs when nucleotides are inserted or deleted from the DNA sequence, causing a shift in the reading frame during translation. This alteration leads to a change in the amino acid sequence of the resulting protein. An insertion mutation involves the addition of one or more nucleotides into the DNA sequence, thereby disrupting the reading frame. As a result, all downstream codons are affected, often leading to a completely different protein sequence. Therefore, option B is correct. A. Substitution mutation. This type of mutation involves the replacement of one nucleotide with another. While substitution mutations can lead to changes in the resulting protein, they do not necessarily cause a frameshift unless they occur in multiples of 3 nucleotides, affecting only one codon. Thus, option A is incorrect. C. Silent mutation. Silent mutations occur when a nucleotide substitution does not result in a change to the amino acid sequence of the protein due to the redundancy of the genetic code. These mutations do not cause frameshifts as they do not alter the reading frame. Therefore, option C is incorrect. D. Missense mutation. Missense mutations involve the substitution of one nucleotide, resulting in the alteration of a single amino acid in the protein sequence. While missense mutations can lead to changes in protein structure and function, they do not cause frameshifts unless they occur in multiples of 3 nucleotides, affecting only one codon. Thus, option D is incorrect. Question 10. Probability for type A blood. 2 individuals, one with blood type 1A and the other with blood type 1B, have a child. What is the probability that the child will have blood type A? A 0%, B 25%, C 50%, 75%. Correct answer, B 25%. Explanation. Blood type inheritance follows Mendelian genetics. Each individual inherits one allele from each parent, resulting in their blood type. The ABO blood type system has 3 alleles, A, B, and O. Alleles A and B are codominant, while O is recessive. In this scenario, one parent has blood type A, genotype AA or AO, and the other has blood type B, genotype BB or BO. When these individuals reproduce, they can produce offspring with blood types A, B, AB, or O. The possible genotypes for the parents are Parent 1, 1A blood type, AA or AO, Parent 2, 1B blood type, BB or BO. When considering the child's possible genotypes, if Parent 1 is AA and Parent 2 is BB, the child will have blood type AB, IAIB. If Parent 1 is AA and Parent 2 is BO, the child will have blood type A, IAIO. If Parent 1 is AO and Parent 2 is BB, the child will have blood type B, IO. If Parent 1 is AO and Parent 2 is BO, the child can have blood type AAO, BIO, or AB, IAIB. Therefore, there's a 25% chance, 1 out of 4 possibilities, that the child will have blood type A. Hence, option B, 25%, is correct. Options A, C, and D are incorrect. Option A, 0%, implies that it is impossible for the child to have blood type A, which is incorrect based on the Mendelian inheritance patterns observed. Option C, 50%, would be the probability if both parents had the same blood type, but in this scenario, the parents have different blood types. Option D, 75%, is an incorrect calculation as it assumes both parents have the same blood type and that the child's blood type is a combination of the parents' types. Yo, it's the med student life, but it's a financial plight. Bills stacking high, feels like an endless fight. Tuition fees looming, like a mountain tall. In this pursuit of knowledge, gotta stand tall. Juggling loans and debts, it's a risky game. In this world of medicine, where's the fame? Stethoscope round my neck, but my wallet's thin. In this hustle for success, gotta find my win. Part-time jobs and side gigs, just to make ends meet. In this financial struggle, gotta keep the beat. Dreaming of the day, when I'll be in the clear. In this medical journey, overcoming fear. In the hustle and the grind, but the finances sting. Balancing budgets, it's a challenging thing. In this world of medicine, where I spread my wings, I'm just a med student, chasing my dreams. Living on ramen noodles and budget meals. In this financial battle, gotta find my zeal. Scholarships and grants, my saving grace. In this journey of medicine, gotta keep the pace. But the pressure's mounting high, like a ticking clock. In this race against time, gotta break the lock. On financial freedom and peace of mind, in this medical marathon, gotta find my kind. Budgeting every penny, like it's my last. In this financial maze, gotta move fast. But I'll push through the struggle, find my way. In this world of medicine, I'll have my say. In the hustle and the grind, but the finances sting. Balancing budgets, it's a challenging thing. In this world of medicine, where I spread my wings, I'm just a med student chasing my dreams. So I'll stay resilient in the face of adversity. In this financial journey, finding diversity. No longer held back by monetary chains. In this medical world, I'll make my gains. Yo, it's the med student grind, but I'll break free. In this journey of medicine, just wait and see. No longer bound by financial woes, in the world of medicine, I'll find my repose.